Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today, the video is related to RNA sequencing data analysis, and the title of my today's video is What is Genome Indexing and How to Perform it Using Linux Operating System. As you all know that I have started a video series related to RNA sequencing data analysis, and this is the part 13th of that video series. And I'm really thankful to you people that you are supporting this series and you are giving a positive response to this series. So basically, I will perform genome indexing in this video. So it will be a real-time practical. So the steps and concepts you are going to learn in this video is that what is indexing and why we perform it, how to retrieve human reference genome for indexing, and how to retrieve human reference genome annotation file. Basically, these are the two files that are needed for indexing. Now then, understanding the base script. First of all, I will explain that before performing indexing, what should be the Linux or the bash script. And after that, we will perform indexing and I will show you the output. So this is a complete video related to genome indexing, and this is very much important. Before starting, I think you all remember the typical workflow for RNA sequencing data analysis. We perform quality control, and in quality control, we do a lot of things. We perform fast QC, fast P, and many other things. So after that, there is alignment. But before alignment, there is a very crucial step that is known as genome indexing. And basically, it is reference genome indexing. So if you are not having the index of your reference genome, so you can never perform alignment. Okay. So because it is included in the workflow of RNA-Seq. So after that, when you get your index genome, then you can align your sequencing reads with the human reference genome, okay, or other reference genome. Okay, so it depends upon you. And we are following RNA sequencing workflow of a human reads. So we will be needing human reference genome. Okay, so before starting with the concept, I just want to discuss and explain an analogy. I believe that explaining a concept using a daily routine analogy makes a person understand well and understand it clearly. Okay, so let's start with an analogy. After that, I will come to the main part of this video, that how to perform indexing. But first of all, you should know that what is basically indexing and why we perform it. So let's start with the analogy. So this is a book. For example, this is a book related to microbiology. And suppose our teacher asks us, or some other people ask us that just search a specific term in this book and or tell me the meaning of that word. Okay, and for example, that word is highly active antiretroviral therapy that is used for treating AIDS patients, okay, acquired immune deficiency syndrome, okay, because this is a book related to microbiology, so there must be some information about the heart therapy, okay. So your first part will be that you have to search that word. Okay, so before searching that word and going through all the pages and finding out that where should the information related to heart is present, so one thing is that you have to go to each and every page and check that where the heart or should be present and where the specific information related to that heart therapy will be present. Okay, but if you are an intelligent person, so you will not follow this step. Okay, you will perform the index step. What will you do? You will just go to the index of this book. Okay, and we all know that in index, this is the index of this book. We will just directly go to the index, and then we will search that my word heart is start from H. So we will go to the H portion. Okay, the H alphabet. Okay, G H. So this is the G and this is the H. And just look here. Here is a word heart therapy. HAART therapy. So you find it out, and after that, it is present on page 931. Okay, so directly you will go to the page 931. We will go to it, and after that, okay, 900. Okay, so this is 931 page, and here must be present the heart therapy. Okay, so yes, this is the heart therapy, highly active retroviral therapy. Okay, so basically in this paragraph, the information related to heart therapy will be included, and you can easily access it, and you can easily tell your professor that what is heart and how it is performed and what is the knowledge related to heart. Okay, so in order you just get that word. After that, you go to the index and you just quickly search out the word, and there it was the page number, and you quickly go to that page number and you just extract that information related to the heart therapy. So basically, this is indexing. Okay, so we are also performing indexing for our genome. Okay, in order to perform active alignment. Okay, or sorry, efficient and quick alignment. So now coming back to the topic, that this Okay, so now we are having a reference genome, and we have performed indexing. So basically, we just index our genome, our entire genome, and we mentioned that where is the specific region, or we are the specific region present, we are the specific sequences present. Okay, and where does that sequencing start from, and what are the actual basis of it. So we index it in an alphabetical order. Okay, so if we are having reads, so if we are not we haven't indexed our genome, so each read will go through all the genome and will find its specific area, and then it will get aligned to it. But when we index our genome, so our read will be quickly analyzed. It will just quickly go to that specific place, and it will get attached, just like the analogy of that book. Okay, so for example, you that her HAART, for example, that is a specific read. And if you're not having an index book, so you must have to go to each and every page of that book, and when you find the information related to that heart term, so then you can read that and you can align your information. But if you're having an index, so you can quickly go to that index and you can just get to that specific word. So basically, this is the main logic behind indexing of your genome. So I think you have get the point that why we perform human reference genome indexing. Okay, so this is the main logic behind it. In order to make it efficient, effective, and computationally less extensive, we perform genome indexing. So there are multiple tools related to that perform genome indexing. There is star tool, bounty tool, okay, that perform genome indexing. Star Solo, Cell Rangers, these are the different Linux operating tools that perform genome indexing. Okay, so we will be using Star Solo or Star tool that is very much effective, and it follows a specific algorithm. Okay, so there must be different principle related to genome indexing where different tools perform different approaches. Okay, there are wheel beaver, roll beaver or reel beaver algorithm. There is a suffix array algorithm that different tools use their own algorithms and principles. Okay, but you have to understand the basic logic of behind genome indexing. So when we perform genome indexing, so we just index our genome, and after that we can easily align our reads to our reference genome. So I think you get the point that before alignment, why we perform genome indexing. Okay, so I think now this what is indexing and why we perform it. I think you get the point, and that analogy might help you a lot in that. Okay, so don't get to the principles of it, that how it is performed and how does the algorithms operate that genome, but just understand the logic behind it. Okay, now for genome indexing, you should have a reference genome and an annotation file. Okay, so a reference genome is basically in FASTA format because it contains the entire nucleotide sequence of the human reference genome. And there is another file that is known as human reference genome annotation file. I want to show you that annotation file. Okay, so this is the general transfer format, GTF file format, that is specific for a reference genome. Okay, so for example, we are having a human reference genome, so it must have an annotation file, because when we are performing indexing, so it is getting the information from that GTF file. Okay, and it is performing indexing. So inside that GTF file, there is a sequence name. It may be chromosome, scaffold, or content, a source name that has been that have generated that sequence, features, the name of that gene, exon or CDS region, start and end, score, strand, frame, and attributes of that specific gene. Okay, so while performing indexing, these two files are important to be present in that very place, and after that we attach these files, then we perform indexing, and a specific output is generated, and that output is used to align our reads. Okay, so this was the main logic behind it. Now I will show you that how you have to get your input files, and the input files are the human reference genome and the annotation file. So let's go to the practical, and I will show you that how to get your reference genome and annotation file. You can also download individual chromosomes. These are the individual chromosomes. We are having these are the 23 chromosomes of the human genome. So you can also extract specific chromosome. Okay, if your supervisor asks you that perform alignment using chromosome 23 or chromosome 1 or 2, so you can easily download it from here. From here you can click on it, or you just click on this chromosome, and after that you can go to the other page, and from there you can download it. So this is human sequence chromosome 1, and you can easily click on the send to. This is the okay, this is send to, this is the file, and you can easily download it in the gene bank format or FASTA format. So gene bank format is basically the full annotated genome, and the FASTA is a simple FASTA, so you can extract it from here. Okay, but we are dealing with the full genome, so we have to download the full genome. Okay, if there is also another database that I liked it very much, and I have extracted the genome from there, it is basically GenCode. Okay, this is GenCode. So GenCode also have reference genome. So this is the human. You can click on it, and after that, now this is here. You can easily download the GTF file and the FNA or the FASTA files. Okay, so this is just look here. It's it is segregated in categories, divided it divided into categories. So this is the first, this is the chromosome. It contains the comprehensive gene annotation on the reference chromosome only, and this is the all the reference, the introns, the exons, and each and everything. Okay, so it depends upon you that which type of stuff you are going to download. Okay, so you can just click on the GTF and it will start downloading. Okay, now it has started downloading, but I'm already having it, so I will just okay, I will pause it. Okay, cross it. Okay, so this is the GTF file, and related to that, these are the FASTA files. Okay, so if this is the protein coding transcript translation, if you want, you are interested in the only the translational regions, so you can download this one. If you are dealing with the all transcripts of the reference chromosome, so you have to download this one. Okay, so basically these are the inputs of it. So I think you understand it well, that how to retrieve human reference genome and how to retrieve the annotation file. Okay, so now you are having the reference genome and you are having the annotation file. So now we will perform indexing. Okay, so let's go to indexing and we will try to perform it. So this is my high performance computing. One thing I want to share with you people, that for human genome indexing, a lot of RAM is used and a lot of cores are being used. Okay, so if you're not having an effective computing system, so it will fail. Okay, it will fail because it required about 8 to 16 GB of RAM and a lot of memory. Okay, so that's why I'm having 125 GB RAM, so it can easily process it. And using this when I was performing indexing, so it took me about 2 hours. Even it was using this all RAM, but it took 2 hours. So I'm telling you that on your computer it may take one night or 6-7 hours to perform complete indexing. Okay, so you should be aware about it, that it required a lot of memory because it is a very big step. Okay, okay, so for human genome indexing, I already have performed indexing, but I just want to share the script with you people, that when you are performing it, so how you will perform it. I'm having my GTF file and my FASTA file or FNF file. So it is about 3 GB of human reference genome and 1.5 GB of GDF file. Okay, so these are the inputs of it, and after that, this is the script of the genome indexing. Okay, so very much easy. I have explained scripts in the previous videos also, so I think you will be aware of it. So Mamba activate star. Well, star is basically the tool that is used to perform indexing. Okay, so first of all, you have to install star tool, and I have already explained that how to install a tool inside your Linux operating system in the previous videos. Okay, so it's also a type of tool. Just install it, and after that, first of all, create an environment and of a specific name, or you can name it indexing or some other name, and after that, install star tool in it. Okay, so that environment will be specific for indexing. So then you will have to activate Mamba activate star, and after that you will perform indexing. Okay, so for indexing, let me show you. Okay, so I'm here. This is the directory. I have made a directory that my output file should be stored in the genome index. Okay, so mkdir, I have made a genome index directory. If you already have made it, so don't just comment out this from the script, that because you already have it. Now you just call star. For the indexing, you choose 24 threads. Okay, so if 24 threads are a lot of threads. Okay, because I'm having a HPC computer, so I have a lot of threads in it, or cores of it. If you are dealing with Core i5 or Core i7, then if you are dealing with Core i5, so you have to give it 4 threads or 3 threads, because it is Core i5. If you are having Core i7, so give it 5 threads. Okay, then run mode genome generate. It should be exactly the same. Genome directory, where is that genome directory? So basically, when we are specifying the output, say that the output should be stored in the genome index. Okay, we already have made it. And after that, this is the genome FASTA files, and this is the input file of the FNF file, and this is the GTF file. So this is the genome FASTA files and the SGB GTF file. So these are the flags, and you have to specify that where my genome file is present. So basically this is the path. Okay, you have to, and this dot hash, this dot slash means that this is my index, this is my script here, and inside this folder exactly these inputs are present. So I just write that I'm present here, so dot slash. If you are performing it in another path, so just give the entire path of your these input files. Okay, so here it will be the entire path, and after that this will be the file name. Okay, so just these are the paths. Okay, so this is also the path, so you can give it your own path. And after that is SGB overhang. This is a complex concept. I'm not going into the detail of it because you will get confused in it. So you have to just write it 99, type it 99. Okay, if you are interested in this concept, so do let me know. I will then explain it in the comments or in the email. Okay, I will explain it to you people, but here I'm not explaining it because it is a complex concept of it. Okay, so this is my script, very much easy. Just go to the directory. For example, I'm okay, cd and human genome indexing, and after that this is the script. So I will just look here. This is the indexing.sh. This was the name of the script. So I will write bash, and after that mm, first of all, before that I will just activate my environment. Mamba activate star, okay, and after that then I will write bash, and after bash I will write indexing .sh, and I will click on enter, and it will start processing. Okay, so I have already performed it. You can perform it on your own computers, but this is the output of it. I want to tell you that when it was performing, so this type of log file was generated. Just look here. I started it on 128, and using the HPC, it was started on 1 o'clock or 1:30, for example, 1:30, and it ended on 3 o'clock, for example. So just look here. It took more than 2 hours to be completed, finished successfully, because it is a very huge step. Okay, so you have to make sure that you are having the exact memory. Okay, so now these are the output files of it. Just look, it is about 30 GB of file, because we have used complete genome of a human. Okay, so this is the 25 GB file, and these are the 1 GB files. So basically it is around about 30 GB file. Okay, so it uses this 4 GB file, and it generated 30 GB file, so after indexing. So just imagine that how much memory it can take. Okay, so this was all about indexing. I think I have impart the complete knowledge about it. The main aim of this video was to explain that what is basically indexing and why we perform it, and I'm pretty sure that you get the point. Okay, if you have get the point, so just do comment in the comment box. Okay, and I also show you that how to retrieve the reference genome in the annotation file. I explain the base script, and exactly I show you that where should you modify it, and then I also perform indexing, or I haven't performed it, but I show you the output files. Okay, so this was all about the indexing. After that we will deal with the alignment. Okay, because performing alignment we will use the index genome as an input. Okay, so that's why we perform indexing. So this was all about today's video. If you have any question, just do let me know. I will answer you in the comment box, or you can also email me about your queries. Or if you are interested in projects related to bioinformatics, and uh, you are facing problems related to it, so do watch my previous video related to how to design a project related to bioinformatics. Okay, it will help you a lot. If you still have any confusion and you are stuck in your final projects, so do contact me. I will help you out, and I will also help you to perform it. Okay, so thank you very much for today. I will meet you in the part 14 of this video series, and will explain the alignment. Okay, then how to perform alignment. Thank you very much. God bless you.